Good afternoon, everybody. Thanks so much for coming, and welcome to our in-person guests today who are enjoying some pizza. And for those of you enjoying this from your office or your home, you're missing the pizza, but we're glad you came anyways. We have a great lecture today, a great visitor. Naiara Aquizu is visiting from Children's Hospital of Philadelphia and the University of Pennsylvania, where she is an assistant professor since, I believe, 2017. She received a PhD from University of Barcelona. She's Basque. And this was in the IBMBC SIC Center on the role of epigenetic chromatin modifications in the nervous system during development, in the lab of Professor Martinez Balbas. And then I was very fortunate to recruit Naiara to my lab for her postdoc, or I say her first postdoc, in around 2011 or so in neurodevelopmental disease, where she uncovered the causes for a number of recessive conditions, including AMPD2 and SNX, with first author papers in Cell and Nature Genetics, describing lots of really interesting new mechanisms. Those were always in collaboration with lots of researchers here on campus. What really set Naiara apart was the thought and care she put into each experiment, always carefully controlled and beautifully interpreted. And she had a CERN fellowship, I think, and a K99, and then spent 2 years in the lab of Yuli Mueller at Scripps to complete her postdoc training and sharpen her skills. And then she and her husband, Joan, moved to Philadelphia, where the Aquizu lab has developed programs in the chromatin repressor EZH family. And she has had several awards, including an Ataxia Foundation Young Investigator, and IDDRC New Program Award, and the Landerbergen Foundation Award. She's also successfully competed for NIH funding with an R01 and R21 grants on the role of EZH in neurodevelopmental disorders. It's really a pleasure to welcome you back to California. We're very sorry about the bad weather today, but looking forward to your talk.
Thank you so much, Joe. Thank you for the introduction and for inviting me here. And as I was saying, I remember my first year. That was very rainy also. So yeah, but good memories. I arrived yesterday with the sun. I saw La Jolla Shore, and lots of good memories came back to my mind. We don't have these nice views in Philadelphia. We have other good things, but not that shore. Okay, anyway, I'm going to tell you a little bit about the work that we've been doing in the lab to try to uncover genetic and epigenetic mechanisms of neurodevelopmental disorders. And as you all probably know, neurodevelopmental disorders are a group of clinically and etiologically heterogeneous conditions that include within the same umbrella rare syndromic cases that are usually associated with brain structural defects or global developmental delays. And then on the other side, more common neurodevelopmental disorders like some autism spectrum disorders or intellectual disabilities that usually affect more clinically defined neurodevelopmental domains. And while the rare syndromic cases are usually caused by single gene mutations, so they are relatively easy to study and find the causes. The common cases are more complex, and they are usually caused by more common genetic variants that lead to the disease only when they converge together within the same individual, or when they interact with environmental factors, leading to polygenic and multifactorial cases that are more difficult to identify and also study in the lab. So despite this heterogeneity, recent advances or last decade advances in next generation sequencing, genome sequencing, and exome sequencing have found several variants that are affected in these patients and several genes that seem to be the cause. And through these studies, we now know that there are some commonalities within all these neurodevelopmental disorders, especially commonalities in pathways and developmental periods that are affected and lead to the disorders. So, for example, a large proportion of variants that are associated with neurodevelopmental disorders fall within chromatin and transcriptional regulators that are usually expressed early during brain development in periods that overlap with neurogenesis, migration, and neuronal differentiation. And then there is another group of genes that are associated with neurodevelopmental disorders, synaptic genes, that are typically expressed in perinatal or postnatal periods. However, for most of these genes and the variants that have been found in neurodevelopmental patients, we still know little about their function in neural development, and even less about the pathogenicity of these variants in these patients. So to try to fill this gap, in my lab, we take genes that have been associated with neurodevelopmental disorders, and we study their influence in neural development at a cellular and molecular level. And through these studies, we have come up with more genes that are involved in neural development, and we have found also new mutations in new genes that we were not expecting associated also with neurodevelopmental disorders. And during these years, we've been mostly focusing on 3 neurodevelopmental disorders caused, each of them by one mutation within one of these 3 genes. The first 2, AMPD2 and SNX14 disorders, were actually genes that we found when I was postdocing with Joe, and I took them with me for my lab to try to study a little bit more the mechanism of the disease and their function in neural development. I'm not going to tell you much about them, but let me just summarize what we've been doing in the lab. AMPD2 is a gene that, when mutated, leads to a disorder that is called pontocerebellar hypoplasia. This is a neurodevelopmental degenerative disorder, and kids with AMPD2 mutations are characterized by having a smaller cerebellum at birth. In addition to the smaller cerebellum, these kids also have degeneration or progressive degeneration of the forebrain regions perinatally, with perinatal onset. And although the mouse model doesn't recapitulate the cerebellar degeneration, it does show a very specific degeneration of the dentate gyrus in the hippocampus. And the work that we've done in the lab has associated the aggregation of IMPDH2 protein in the form of these very particular aggregates with rods and rings form in the degeneration of the dentate gyrus in the mouse. And I think what is even more interesting is that a couple of years ago, there were patients with neurodevelopmental disorders that were found to carry mutations in IMPDH2 gene. And a recent study from Justin Coleman's lab in the University of Washington has identified that some of these mutations actually affect the aggregation or polymerization of IMPDH2. I'm not going to tell you much about this, and I'm going to just summarize also what we found with SNX14. SNX14 is also a similar disorder. It's also a neurodevelopmental degenerative disorder that affects the cerebellum. And with a mouse model that we generated, we have linked this cerebellar degeneration with lipid homeostasis and clearance in the cerebellum and ferroptosis. But what it is even more interesting is that through the studies that we were doing in the lab to try to understand the function of SNX14 in neurons, we found that SNX14 interacts with SNX13. SNX13 is a very similar protein, and they both together are also involved in lipid homeostasis. This is a study that we are finishing now. But again, the most interesting is that as we found this mutation and started working on the function of SNX14-13 interaction, we were contacted by a clinician that had a patient with a clinical feature or a clinical disorder very similar to the patients with SNX14 deficiency, but this patient had a mutation, an homozygous nonsense mutation in SNX13, suggesting that SNX13 is also leading to a similar disorder of SNX14. We've been trying to find more patients with SNX13 mutations. We haven't been very successful. There is just one report for a year ago describing one single patient with a very similar mutation in SNX13 and also cerebellar degeneration with intellectual disability syndrome. Okay, So what I'm going to be focusing today is in the third story, a story that started, I think, I would say mostly by accident, from some data that I generated in my PhD when I was trying to uncover the function of epigenetic modifiers or chromatin modifiers in neural development. And now this sounds obvious, right? But back then, when I was doing my PhD, this was right here, right before the implementation of next generation sequencing methods that have promoted all these sequencing initiatives in patients, in many patients, including neurodevelopmental disorders, and have led to identify that chromatin mutations are abundant in these neurodevelopmental disorders, suggesting that chromatin is important for neural development also. But back then, when I was doing my PhD, the only association between chromatin and neural development was probably mutations in MSCP2 that could also be found in Rett syndrome a few years before I started my PhD. However, at that time, we were starting to learn that chromatin modifiers were important for early embryonic development. And we were also starting to learn that chromatin mutations in chromatin modifiers were involved in cancer also. So my working hypothesis during my PhD was that chromatin modification and chromatin modifiers would be important regulators of neural development. And to study this and to test this hypothesis, the chick embryo model was a great model organism, especially in Spain in a small lab that I was doing my PhD, because it is an affordable model first, and second, the embryos are easily accessible. You can just open the egg and you have the embryo right there. And in addition, the chick embryo neural development recapitulates many aspects of mammalian neural development, including the temporospatial segregation of different neural types during development. And this is best illustrated here. This is a cartoon of a neural tube transversalization, chick embryo neural tube transversalization. It looks very similar to mouse or human neural tube. And in this neural tube, we can differentiate a ventricular zone around the neural tube lumen. This ventricular zone is full of neural progenitors that divide and expand the pool. And then as soon as they start differentiating, they migrate to the periphery and they do their way there in the periphery in the mantle zone. So in order to test if chromatin modifications and chromatin modifiers had a function in neural development, this was a great model because I could just stain for chromatin modifications and chromatin modifiers and check what was the pattern of expression. And among different tests that I did with different antibodies, I found ECH2 and ECH1 expression pattern one of the most interesting. As you can see here, ECH2 is highly expressed in the ventricular zone where all the neural progenitor resides. And then as you go to the periphery where the neurons reside, the expression level is lower. While ECH1 has the opposite expression, with low expression in the ventricular zone and high expression in the mantle zone where neurons reside. So to test if this expression pattern was significant functionally, the next thing that I did was to manipulate the chick embryo neural tube genetically by electroporating DNA plasmids into the right side of the neural tube, into the neural progenitors of the right side of the neural tube, and then monitor how they differentiate and how they develop by the co-expression with GFP. And as you can see in this just one experiment that I did, while the electroporation of control plasmids leads to a segregation of electroporated neurons or neural progenitors between the ventricular zone and the mantle zone, and the neural tube look the same, like the electroporated side and non-side look the same. When we knocked down ECH2, the first thing that we saw was that the neural tubes were much smaller. In addition, TUCH1 expression, which is a marker of differentiated neurons, was now in the ventricular zone where neural progenitors usually reside, so unusual expression. And these TUCH1 positive cells were the electroporated cells, so cells that had less ECH2, indicating that ECH2 is necessary to maintain neural progenitors proliferating, and when you knock down ECH2, neural progenitors differentiate right away. I spent my PhD working on ECH2 mostly But what I found more interesting was the effect of ECH1 knockdown. When we knock down ECH1 in neural progenitors, the neural progenitors got stuck in the ventricular zone, unable to differentiate and migrate to the periphery, which led to less neurons in this electroporated neural flux. However, at this time, there was not much known about ECH1, pretty much anything, and the only thing that we knew was that ECH1 was an unfunctional copy of ECH2, so no one was paying attention to ECH1 even. Now we know a little bit more. So what we know right now is that both ECH2 and ECH1 are the methyltransferases of the polycomb repressive complex 2. And within polycomb repressive complex 2, ECH1 and ECH2 mono, di, and methylate lysine 27 of the histone H3. And this is a chromatin modification that is involved in the epigenetic transmission of the transcriptional repression. And this epigenetic transmission of the repressive state is important for lineage specification and maintenance, and also for the maintenance of cell identity in the adulthood and also homeostasis. However, most of what we know about PRC2 come from studies focused on ECH2. First, because ECH2 is still the most catalytically active copy. The ECH2 is about 10 times more catalytically active than ECH1. Second, because ECH2 is also highly expressed in proliferating cells that most of us use in the lab to study, so it's easy to work with. And third, the significance, the disease relevance of ECH2 has been known for a long time. Almost 20 decades ago, it was already found that overexpression of ECH2 was typical in many cancers, including the first one was prostate cancer. And then in the last decade, there have been like many mutations in ECH2 that have been found in cancers, mostly in lymphomas. 20% of lymphomas have ECH2 mutations. And a little bit more recently, mutations in ECH1, de novo mutations in ECH1 have been also associated with Weaver syndrome, which is overgrowth with intellectual disability syndrome. So what was my surprise when in 2019, in just a random conversation with Elizabeth Bosch, a clinician scientist at CHOP, I found that she had a patient with a neurodevelopmental delay, with intellectual disability and dysmorphic faces, with a mutation in ECH1, a missense mutation that was affecting evolutionarily conserved amino acid that is also that has very little variability in humans. And when we modeled this substitution, the leucine to phenylalanine substitution in ECH1 structure, what we found was that the phenylalanine ring was generating a stacking interaction with the adenine group of the SAM methyl donor in the catalytic pocket of ECH1, suggesting that this stacking interaction could be affecting the catalytic kinetics of ECH1 and therefore suggesting a pathogenic effect of this mutation. This was 2019, and since then, by word of mouth and matchmaking initiative, we've been able to expand this cohort to 19 patients that we published recently, and then I think almost every month we get a new patient. Now we have 4 more patients that have mutations in ECH1. All of them have intellectual disability or developmental delay, including also delays in motor development, language, and social development. And then most of them have unremarkable findings in their brain imaging, with the exception of some of them that have mild white matter loss, or this one here that has a little bit more severe white matter loss and ventriculomegaly. This is probably one of the most severe cases. Pretty much all the other patients have unremarkable brain structure. And then most of them have mild dysmorphic faces, also like mild deep set eyes or mild lower ears, and some of them have also prognatism. At the genetic level, these mutations are also classified in 2 different groups. Half of the patients have heterozygous missense mutations in ECH1. And one interesting thing that we are starting to learn now is that half of these mutations cluster within the set catalytic domain or the around the set catalytic domain, while the other half of the mutations are clustered in the MCSS SANT2 loop that has been recently identified as a region of ECH1 that is involved in, that participates in the interaction with nucleosomes and also in the compaction of chromatin. And this is a function that is exclusive of ECH1 and not present in ECH2. And it's also a function that is independent of the lysine 27 trimethylation activity. I will go back to this a little bit later, but I want to tell you a little bit more about the other half of the patient. The other half of the patients have all biallelic mutations or homozygous mutations that lead to ECH truncation, with truncation effects predicted to lead to loss of ECH1 expression. And just to validate that this prediction was real, we were able to obtain cells from this patient that has a deletion of 5 exons in one allele inherited from the mother, and in the other allele, the novel mutation that affects the splicing. And by RT-PCR, we validated that the exon 10 was excluded in the transcripts of this patient. And then by Western blot, we also validated that the expression of ECH1 is completely gone in the patient cells. Just also to check that the nonsense mutations lead to loss of function, we generated cells from this nonsense mutation in homozygosity, and we also found that ECH1 levels go down. So we just wonder whether the loss of ECH1 in human brain would have an effect similar to the ECH1 knockdown that I found during my PhD. So basically, would ECH1 loss impair neuronal differentiation? That was our question. And to answer that question, we moved to human pluripotent stem cell model. From human pluripotent stem cell, we can make neurons and we can make organoids of all types. One of the experiments that we did in the lab was to generate forebrain organoids from human pluripotent stem cells. And similar to the chick embryo, if you take these organoids and you section them transversally, you can see lots of these neuroepithelial structures that are organized in a similar way, with a ventricular zone full of neural progenitors, and then as neural progenitors divide and differentiate, they go to the periphery. And when we stained these organoids with ECH2 and ECH1, we also saw a similar expression pattern, with ECH2 highly expressed in the ventricular zone, and then low expression in the periphery where neurons reside, while ECH1 was quite homogeneous, both in neural progenitors and also in the neurons. And that recapitulates also in the human brain development. This is data from BrainSpan, expression data from RNA-seq. And as you can see here, ECH2 levels are high early during brain development, but as soon as neurogenesis proceeds and neurons start differentiating, ECH2 levels go down, leaving ECH1 as the predominant lysine 27 methyltransferase, starting from neurogenesis to the adult brain also. Okay, so we next generated ECH1 knockout pluripotent stem cells, just with CRISPR generating random mutations. These stem cells don't express ECH1, and then we made forebrain organoids, 60-day-old forebrain organoids. And as you can see here, wild-type organoids are composed by neural progenitors labeled with SOX2 here in the middle. And then in the periphery, we see mostly CTIP2 early-born neurons. There are few of the SATV2 type of neurons also, but this is still early for these neurons to show up. When we compare these neuroepithelial structures with the ECH1 knockout organoids, you can easily see that neural progenitors are still there, but now we lose neurons. We lose the early-born CT2 positive neurons, recapitulating actually what we found in the chick embryo. Basically, ECH1 is necessary for neurons to differentiate, and ECH1 knockout brain or ECH1 knockout brain organoids are unable to differentiate neurons, or they have a delayed differentiation process. So most of the efforts in the lab right now are focused on trying to understand how molecularly ECH1 regulates this neuronal differentiation. And organoids are great to study phenotypes in the brain or in neural development, but they are very heterogeneous cellularly. So all these like biochemistry techniques or molecular techniques that we do are not very useful in organoids due to the cellular heterogeneity. We can minimize a little bit the cellular heterogeneity by differentiating pluripotent stem cells to neurons in 2 dimensions. And that's pretty much what we are doing now to try to understand the function of ECH1. And as you can see here also, when we do this differentiation, ECH1 expression is quite homogeneous from pluripotent stem cells to neurons. These are 4 we call neurons, while ECH2 is high in all proliferating stage, progenitors, neural progenitors, but as soon as we start differentiating these neurons, ECH2 levels go down. So the first thing that we did in the lab was to take these neurons that are cellularly or cell typely more homogeneous and don't have ECH2, so we can extract the function of ECH1 without having ECH2 there overlapping or masking ECH1 function. And with this, we have done some RNA-seq. In the RNA-seq data, we see similar phenotypes. Neurons that don't have ECH1 have higher expression of proliferation markers or neural progenitor markers, and they have lower expression of late-born neuron markers than the ECH1 wild-type cells. And I'm not going to show you much more, but what we are trying to understand now is how ECH1-derived lysine 27 trimethylation is involved in these changes. And for this, we are trying to still make sense of this data, but we have done an analysis of lysine 27 trimethylation distribution in genome-wide. And from this data, what we found was that there are not many changes between wild-type and knockout neurons. And if there is anything, actually, knockout neurons have a little bit more lysine 27 in coding regions, at least in the peak and in the spreading part. So our model, based on these phenotypes and this very preliminary data, is that ECH1 relieves ECH2 repression on neuronal differentiation genes. And let me just explain you this briefly. In neural progenitors, where ECH1 and ECH2 are expressed, ECH2 is trimethylating and silencing neuronal differentiation programs. And as neurons differentiate and ECH2 is lost, then we just have ECH1, which is 10 times less catalytically active, so can't maintain those programs silenced, and they are open to be activated and push neurons to differentiate. Instead, in the ECH1 knockout cells, ECH2 will replace the localization of ECH1, leading to more methylation. And during the differentiation, this increased methylation will inhibit the reactivation of differentiation programs. And that results in delayed differentiation and in a slightly higher levels of lysine 27 trimethylation that we see in the neurons. Still lots to do. We are trying to find where ECH1 is, where ECH2, and how the dynamic process from neural progenitors to neurons happen, because I think just looking at the neurons, we are missing a lot of information, but I hope I can tell you this next time I can. Now, what I'm going to tell you is a little bit more about the other type of mutations. So what is the effect of the gain of function mutation? Oh, sorry, heterozygous missense mutation, spoiler there. So what is the effect of the missense mutations? Just by looking at them, it's difficult to predict what the missense mutations do, right? But knowing that the clinical features of the patients with the missense mutations are similar to the patients with biallelic loss of function mutations, and also knowing that losing one copy only, it's not enough to cause a disease. We initially thought that the missense mutations would act like a dominant negative mutation. So to test this, we took some of these mutations, particularly these 3 mutations that are localized in the Z domain or around the Z domain. And also these were the first 3 mutations that we found in our cohort. So we took these 3 ECH1 variants and we ectopically expressed them in a neural progenitor cell line. And what was our surprise, that opposite to what we thought, we found that the overexpression of these variants led to increased lysine 27 trimethylation. And we were not still convinced. We thought that, well, ECH2 is still there, and maybe ECH2 is doing something and masking our phenotypes. So let's try to find a model where we can remove ECH1 from the picture. We can remove ECH1 for our equation. And we found this model basically in one of the assays that Danny Reinder does all the time in his lab. So we contacted him and we did these histone methyltransferase assays in his lab. So in this experiment, we incubate recombinant PRC2 with wild-type ECH1 or with one of the 3 variants, together with unmodified nucleosomes or the predefined dimethylated nucleosomes. And in these reactions, we also add radioactive SAM, radioactive methyl donor. And after incubating these 3 components together, we measure the level of methylation of the nucleosomes, which is proportional to the autoradioactivity signal that we see in these plots here. And just to summarize, what we found from this experiment is that, yeah, all the mutations are leading to more catalytic activity, but in a slightly different way. While the L2F mutation increases slightly and actually not very significantly the methylation of unmodified nucleosomes, it doesn't really have much effect in the dimethylated nucleosomes. Pretty much as the wild type, we don't see much methylation with the L2F mutation. The Q2E mutation leads to increased methylation on unmodified nucleosomes and also in the dimethylated nucleosomes here. And then the A2G mutation is probably one of the most interesting because, as you can see here, this mutation leads to increased methylation of dimethylated nucleosomes, but is completely unable to methylate unmodified nucleosomes. And this is also actually reminiscent of many ECH2 cancer mutations. There are several cancer mutations that have been characterized molecularly, particularly this Y2N mutation. This is quite abundant mutation. It's present in more than 20 or around 9 to 20% lymphomas and some melanomas also. And then some patients also have this A2G mutation, which is actually the mutation that is equivalent to the ECH1 A2G mutation. It's pretty much exactly the same. And in 2020, Maccabi and his colleagues tried to characterize the effect of this A2G mutation. And what they found by modeling the structure is that the glycine increases the pocket of the lysine, accommodating better the dimethylated lysine and making the unmodified lysine less stable in the pocket. And this effect leads to increased or a better methylation of dimethylated lysine rather than unmethylated lysine. And that's the opposite of the wild-type ECH2. Wild-type ECH2 is much more effective monomethylating rather than trimethylating. So in combination in heterozygosity, like in cancer cells, the combination of wild-type ECH2 together with mutant ECH2 leads to mono and dimethyl and then more trimethylation, as you can see here in these lymphoma cell lines that have all these ECH2 mutations and increased lysine 27 trimethylation. And these findings have motivated the development of a large repertoire of ECH2 inhibitors. Some of these are already, some of these are in clinical trials, others are approved. I think this one is approved for epithelial sarcoma, and this other one that is ECH1 and 2 dual inhibitor is approved for B-cell lymphoma, some aggressive B-cell lymphomas. So this encouraged us to explore if the repurposing of these drugs could be eventually useful to treat our ECH1 gain-of-function patients. And we have just started testing this, but what we know is that it's very difficult to remove lysine 27 trimethylation from neurons that don't divide for some reason that I'm not going to get into detail now. But we were able to reduce lysine 27 trimethylation a little bit in some immature neurons with the treatment of this dual inhibitor. We are now trying to see if some of our phenotypes are also improved. But before I go there, I want to tell you that this dual inhibition is probably not a very good idea as a treatment, as a chronic treatment for patients with neurodevelopmental disorders, because from some work that has been done in the past from Anne Schaefer's lab, for example, we know that deleting postnatally ECH1 and ECH2 leads to degeneration, to neurodegeneration. These are cerebellar sections of mouse that doesn't have ECH1 and ECH2 in Purkinje cells, and you can see that at 6 months of age, there is a loss of Purkinje cells in these brains. They have also tested this in other brain regions, and it's pretty much the same. Also, a recent study from Lorenz Struder lab shows that inhibiting just transiently ECH2 in neuronal differentiation just early on leads to more mature neurons. So based on these two evidence, we have embarked in the adventure of trying to develop ECH1 selective inhibitors. And in order to achieve this goal, we have partnered with John Nicholas, who has a lab in the Fox Chase Cancer Center dedicated to develop computational approaches to identify cryptic druggable sites in the surfaces of proteins, and then to screen for small molecules that can engage into these cryptic sites and inhibit targeted proteins or proteins of interest. So we started using this approach to identify ECH1 selective regions that we could use to inhibit ECH1 but not ECH2. But unfortunately, the set domain, the catalytic domain of ECH1 is very similar in structure and in sequence to ECH2 sequence, and we were unable to find any site that was specific for ECH1. The only alternative that we found was this cysteine that is localized close to the active site, close to the set domain of ECH1, and it's not present in ECH2. And we could potentially use covalent inhibitors to engage this cysteine and block the catalytic pocket of ECH1. But we know that covalent inhibitors are very reactive and they have lots of side effects, so it's not an alternative for therapy anyway. So as a third alternative, we have started to explore another, like a completely new thing, pretty much inspired in the new class of molecular glues that can target and degrade pathogenic proteins by recruiting them to E3 ligases and promoting their ubiquitination and proteasomal degradation. There are several examples out there, but not very many, pretty much molecular glues, few molecular glues that are currently used for the treatment of cancers. And molecular glues are good because they are usually small, so they have a good bioavailability and good cell penetrance, and that means that we can easily potentially bring them to the brain also. They are also easy to synthesize. But the problem with molecular glues is that they have unpredictable design. So far, all the molecular glues that are out there have been found by serendipity, not for rational design. However, with the expertise of John Nicholas in protein structural modeling and in small molecule screening, we have designed a strategy that we hope we can successfully apply to find ECH1 specific molecular glues. And this strategy is based in 4 steps. In the first step, we will use machine learning approaches to identify E3 ligases that interact with ECH1 in a real way or in a functional way. In the second step, we will identify pockets or regions between ECH1 and E3 ligase structure that fit small molecules, and then we will screen small molecules to engage to these regions and keep the 2 proteins together. And then in the third step, we will validate the small molecules experimentally, and in the last step, we will characterize the molecular glues. Right now, we have already completed the first step. For the first step, we designed a machine learning approach, and to design this machine learning approach, we took experimentally confirmed E3 and substrate structures, and then we scrambled them and generated AlphaFold models of both of the experimentally tested and scrambled pairs. And then we extracted the structural features that predict the real or experimentally validated interactions to train a machine learning classifier to identify real interactions. In the second step, we built AlphaFold models of ECH1 with 32 different E3 ligases for which a structure is already known. And then we put these structures through the machine learning classifier to identify the interactions or the pairs that had a real or that were predicted to be functional. And from this screening, we found 5 of these E3 ligases that have good scores for interacting with ECH1. And here I'm just going to show you one of these examples to illustrate what a good interaction or a real interaction means. In this case, we have BTRC, E3 ubiquitin ligase, in gold, and the interaction with its endogenous substrate, beta-catenin, in red here. And if you look at the AlphaFold model that was selected by the machine learning classifier, the structure of BTRC in green now overlaps pretty much with the endogenous structure of BTRC. And the interaction of ECH1 happens at the same surface as the endogenous beta-catenin with BTRC, suggesting that the geometry of the interaction, which is important for the ubiquitination to happen, is maintained in this structure, and suggesting that this is a promising E3 ligase that can degrade ECH1. So instead of going directly to the small molecular screening, we started thinking on what are the experiments that we can do to validate that these ubiquitin ligases can actually degrade ECH1. And this is not easy for several reasons either, but one of the approaches that we did was to overexpress these E3 ligases in 293Ts first and then in human pluripotent stem cells. And from these overexpression experiments, we found that the overexpression of BTRC consistently reduced ECH1 levels, both in 293Ts and also in pluripotent stem cells, with some variabilities, but the reduction is consistent. So our focus now is on BTRC ECH1 interaction, and what we are doing is screening small molecules computationally using a database of 21 billion structures that John has in his big computer in the lab, and screening how these molecules can interact between in the surface between ECH1 and E3 ligase. And from this, we hope to get around 40 compounds that we can validate experimentally for ECH1 degradation, and then also for for testing our phenotypes in vitro. And before I tell you what are the phenotypes that we found, that I didn't tell you yet, I just want to let you know that our motivation to generate these inhibitors or these small molecule degraders goes beyond the ECH1 gain of function associated neurodevelopmental disorder, and have maybe broader applications, including applications in CAR T-cell generation or CAR T-cell therapy. And this is based in a couple of publications from George Daly's lab that found that ECH1 inhibits lymphoid cell lineages in embryonic hematopoietic stem cells. And then a follow-up paper showing that transient inhibition of ECH1 from human pluripotent to T-cell differentiation enhances the maturation of these T-cells in vitro. And actually, these T-cells engineered with CAR-Ts have much powerful effect in mice than the regular T-cells that are differentiated from iPSC. So we could potentially use this drug also to make T-cells in vitro and then improve CAR T-cell therapies. But going back to ECH1 gain of function disorder, the first question is whether we can actually treat neurodevelopmental disorder, right? Can we really rescue a neurodevelopmental phenotype that was induced early on during development? And probably I don't need to convince you guys about this. I don't need to convince this audience about this, but there are actually several examples in the literature showing that postnatal treatment or postnatal rescue of developmental insults improves some cognitive function, right? So there is, for example, the recovery of UB3 in Angelman syndrome has been shown to improve cognition in mice. Also, for the MECP2 duplication syndrome, There are examples showing that reducing MECP2 levels postnatally can improve cognitive functions, and Kabuki syndrome, which is another syndrome associated with chromatin modifications. So in Kabuki syndrome, also treatment of these mice postnatally can improve some cognitive functions. So what about ECH1 gain of function neurodevelopmental disorder? Can we treat this neurodevelopmental disorder? In order to test this, like we did with ECH1 loss of function mutations, we also introduced ECH1 gain of function mutations in embryonic stem cells. And in this case, we introduced mutations in H9 embryonic stem cells. And then in order to validate our phenotypes in a different genetic background and in a male background, we started using KOLF 2.1J iPSCs that were reported a couple of years ago to be very efficient with CRISPR editing, and also good for differentiation to different neuronal lineages. And they are actually great for CRISPR editing. We were able to make lots of mutations in several months. However, we found a problem. When we started validating all these mutations, what we found was that all these edited lines had several copy number variants that were not reported in the original paper, where they did also genome sequencing and SNP arrays to validate the genome. But these variants were not reported there. So we were not sure if these copy number variants were only in our clones, or they were also present in all the KOLs that were out there. So to validate this, we went to the genome sequencing data that they published in the paper, and we analyzed these regions. And actually, as you can see here, for one of these copy number variants, the number of reads in this region are lower, and there is a loss of heterozygosity, also confirming that these copy number variants are actually present in the original KOLF cell line. And maybe we wouldn't worry too much if it wasn't because one of these copy number variants falls within JARID2. Actually, deletes almost all the exons of JARID2. So what is JARID2? JARID2 is actually an accessory subunit of the PRC2. It's a subunit that is necessary to improve the interaction of PRC2 with nucleosomes, and it's also a subunit that enhances the catalytic activity of PRC2. So probably not a good idea to use this cell line to study ECH1. But in addition, it's already known that haploinsufficiency of JARID2 leads to a neurodevelopmental disease. Haploinsufficiencies that are similar to the copy number variants that we found in KOLs, but also lots of patients with small deletions or nonsense mutations in JARID2 have neurodevelopmental disorders. So we thought that probably modeling a neurodevelopmental disease over another neurodevelopmental disease was not the best idea, and we took all these edited lines and we froze them down and we have them in the liquid nitrogen, just thinking on what we can do with all these edited embryonic stem cells. But let me go back to our original H9 embryonic stem cells that we edited with this A2 gene mutation. So similar to what we did with the loss of function variants, we generated 4 brain organoids for these A2 gene mutations. And as you can see here, the phenotype was actually the opposite to the phenotype that we see with ECH1 loss of function organoids. In this case, you can see that the amount of SADB2 positive neurons is large in the ECH1 gain of function for brain organoids, indicating, together with several other experiments that we did, that ECH1 gain of function mutations accelerate neuronal differentiation. And like for the loss of function mutation, we are trying to understand how molecularly this happens. And in this case, we also analyze the lysine 27 levels, lysine 27 distribution in the genome in wild type and gain of function neurons. And in this case, we saw that actually the gain of function neurons have increased levels of lysine 27 trimethylation in coding regions. And when we take these regions, the regions that are gained, the lysine 27 trimethylation regions that are gained in the ECH1 gain of function neurons, and cross them with genes and then analyze what's the expression of these genes in these neurons. What was our surprise? That what we found was that there were lots of genes that had higher lysine 27 trimethylation and were expressed at lower level in these neurons. genes that are involved in synaptic activity, glutamatergic synapses. So again, now the opposite, indicating that even though gain-of-function mutations lead to an accelerated differentiation, the neurons that are generated from this differentiation are not functional. But the good thing about this is that if this is true, if these neurons are there but are not functional due to the higher lysine 27 trimethylation, if we can inhibit postnatally ECH1 function, or if we can degrade ECH1 and reactivate those genes in neurons, then this is a good therapeutic approach. So we still need to do a lot of work to understand the molecular mechanism of all these changes. But from these experiments, we think that investing a little bit of time in developing these inhibitors or molecular glues can have important effects in the future. And with this, I just like to summarize, showing a little bit or summarizing a little bit what we have done in these years and what we are hoping to do in the future, starting from genes like AMPD2, SNX14, and ECH2, with the functional work and the biology that we do in the lab, we've been able to identify new disease variants and also potential therapeutic approaches. And with this, I would like just to acknowledge and thank you for all the work that all the clinicians and geneticists that we collaborate with do for us. And also all the other collaborators like Danny Reinberg and many more that have been helping us with our studies. And then a big thank you also to my lab. This work was mostly done by Carolina, that just graduated on November, and now she's starting a postdoc in JSK in a company there in Philadelphia. And a big thank you also to everyone else, especially to Thomas, that has been a game changer. Thomas is a postdoc in my lab that started a year ago, and he's fully computational. So he is starting to make sense a little bit of all these cat and runs and RNA-seq data sets that we generated in the last 2 or 3 years. So with this, I would just like to thank you everyone for coming, and if you have any questions, just please go ahead.
Thank you very much for your presentation. Very interesting. I was wondering, do you have a plan or have you had any single-cell RNA sequencing on your organoids, especially during the development?
Yep.
Following up like different cells out of the response.
Definitely, that's something that it's ongoing. Yeah. Mm-hmm.
And I actually have already different question. I don't even know whether it makes sense, so you tell me. Have you ever tried to check whether you show us among all of your patients that have different mutations that happen in different areas of these genes with different mutant tolerances? Mm-hmm. Do patients that have mutations in less tolerable areas represent stronger phenotypes, both functionally or structurally?
Not really. No, we have looked at this, and it's actually a little bit surprising because one of the patients that have a mutation in the less tolerable area has one of the most severe phenotypes. And I was telling you before, this patient also has a variant of significant unknown significance in another chromatin regulator. So we think that the combination of the two variants is leading to more severe phenotype. Yep.
One, do you consider ASO targets?
Discussing with Joe. Yeah. Yeah, definitely.
The other one is more molecular. Are there clues, or do you have clues on how the regulation of changes with like differentiation? What makes it go down when they?
No, that's a very good question. We don't know that. There was a paper from Struan and Hopkins lab in blood cell differentiation involving GATA2, I think, in the silencing of ECH2, I think, something like this. But this, I think, in my understanding, are very specific of blood cells, so I'm not sure if the same factor will play a role in silencing ECH2. But yeah, that's interesting. Mm-hmm.
First, thank you for this great talk. So I'm trying to understand the logic of getting rid of one altogether, because in the beginning you said that the overall methylation will not change because EZH2 kicks in and then it doesn't. And then the second you pointed out that this different mutations generate different selectivity versus unmethylated mono and di, right? So how does, like, if you have some like a perfect degrader, right? So how does it help you? Because it's you know, this previous two statements basically say that it's too blunt of an instrument.
Yeah. So let me see if I understand properly. What I think you are saying is, like, if ECH1 loss leads to the disease, how removing ECH1 from the gain of function is going to help to the disease, right? Because we are also reducing ECH1.
Because
right?
you're saying that the EZH2 is still there, and it's 10 times more active.
Yeah. The thing that we have to take into account, and I think maybe I didn't mention this, I didn't highlight this. What we think is that the ECH1 loss of function patients, their disease is more developmental, so there are no neurons that are generated, right? So even if we treat or do something postnatally, I don't know if we will solve that problem. For ECH1, we make neurons, and we are not going to touch the development. So we are not removing ECH1 during the development. We will remove ECH1 postnatally in the neurons that they already don't have ECH2. So that's a little bit what I think it's going to help those patients. And also the idea is not to get rid of ECH1, because I don't think that getting rid of ECH1 completely will be helpful anyway. There is a report there showing that the reduction of ECH1 is associated also with schizophrenia postnatally. So I'm not very confident of that word, but you know, it's possible that removing ECH1 from neurons can also have other side effects. The idea is that with these small molecules, if they work, we can treat the patients, and we can maybe not control the dose very well, but we can keep the dose in a level where we can still maintain some ECH1 active, or if things go wrong, remove the treatment and get more ECH1 to do their job, right? So that's a little bit the idea of the small molecules. But the question, going back to your question, I think the key is when we remove ECH1. So removing early on when ECH2 levels are high has some effects. Removing later on when there is no ECH2 has other effects. And I think that's the key that we need to understand.
Can you ask a follow-up?
Yes.
Yeah. Okay, I can ask you. You showed that there was no change in the overall trimethylation in the EZH1 knockout, but and you think that's because EZH2 is compensating. But if you look at the gene specific, I mean, do they really have the same? Because there's no there are some synaptic proteins and things like this. So the overall level might be the same. The transcriptome might be very very different, right?
Yeah, completely. And that was average of all the genes, right? So what are the genes that are targeted by ECH2 versus the ones that are targeted by ECH1? That's something that we are working in the lab. Challenge there. There are no good antibodies for ECH1 cut and runs or ChIP-seq. We have tried alone. We have found one that looks like it's working. At least it's working when we overexpress ECH1, and we have done some cut and runs in neurons that have slightly more ECH1 also. And if we compare, the signal is much lower, but if we compare the signal of the overexpression with the wild type ECH1s, they overlap. So that's just a very recent result from a month ago. We think we have the tool now. We can't make any conclusion if we don't know what ECH1 is. Yeah. Yeah, go ahead. Maybe we can do two more questions, Joe, one on one. I will try to be short.
You're very generous. With asking you, so my question is, if one is two is out of picture, then why don't you use small doses for the approved 1 and 2 inhibitors?
Yeah, that's a good suggestion. Yeah. Definitely.
I mean, why bother?
Yep. Yeah. No, definitely, and that's probably closer to the succeed than the molecular glues approach. Definitely. Yeah. Thank you.
So it may help with the.
Yeah.
You try to do a comparison
Yes. It's completely different. So that's the first thing that we did. We did cut and runs in the wild types, knockouts, and the gain of function mutants. And we thought, okay, so we don't have ECH1 antibody, but it's going to be easy. We just need to compare which genes go down, lose lysine 27 trimethylation, which ones gain, and this will be ECH1 targets. There are maybe like 5 or 6 genes that go down and up between the knockouts and the gain of function, but easy explanation. We are doing all this in neurons. These neurons have lost ECH1 months ago and have also gain of function mutations months ago. What we are comparing are apples with pears, pretty much, and that's most of the RNA-seq and cut and run experiments that we are analyzing. So we need to do this in a slightly different way.
You should also consider
we are using a spike in, we're using the cut and run spike in nucleosomes also, but even with the normalization, I think it's a problem of the cell types that we have rather than normalizations. So probably what we need to do is make neurons first and then knock out ECH1 and overexpress ECH1 or something like this. Yeah, not from the beginning. Yeah, or do this in other cell types that are not neurons. Okay, thank you so much, everyone.